everyone, today we're going to be talking about vaccines, specifically the vaccines that protect us against viral infections. And basically the role of a vaccine is to induce a quick and secondary response to infection that involves the production of antibodies and also the cytotoxic T lymphocyte response. And we actually have some natural sources of vaccines, such as the ones that we get from our mothers when we're breastfed. It's a natural passive vaccine because it induces an IgM response. And also when you're delivered as a baby, the vaginal secretions can also protect you. Because it has some bacteria as well, and it acts as a natural source of immune stimulation. And I guess with vaccines you can go several different ways. You can just attenuate it, meaning that it doesn't infect you and doesn't cause disease. You can inactivate it. You can fractionate it or break it apart and use the subunits of the viruses, or you can clone the viral genome and then manipulate this to create virus-like particles. And today we're going to be talking about the advantages and disadvantages of each technique. But before we do, let's talk about smallpox. So smallpox is a disease with a high morbidity and mortality. So it's important to eradicate it because it has a huge economic benefit. And basically the eradication of smallpox has been successful, and we believe that this might be due to the fact that it is not a very persistent infection. It has very visible symptoms. It doesn't have any secondary hosts other than humans. It has a long incubation period and a low communicability. And also it only becomes infectious after the incubation period, unlike other diseases where you can be asymptomatic but still be able to transmit to others. And basically the vaccine for smallpox evolved. Firstly, we had like a variolation inoculation of the smallpox pustule, and this was done in China and India way before the discovery of viruses. Then Jenner found that if you put cowpox into humans, then they gain protection against smallpox. And this is because cowpox just infects cows, but it doesn't infect humans. But perhaps the immune response against cowpox is just as effective in protecting against smallpox, and the immune memory established from that is quite useful. Nowadays, we use the vaccinia pox virus. They have very little genetic similarity to the smallpox and the cowpox, but still it garners the necessary protection. Now the next viral infection that we are almost done eradicating with is poliomyelitis. So now it is only found mainly in Afghanistan and Pakistan. And we owe this dramatic drop in poliomyelitis, which can cause paralysis, and in half of all survivors, and causes death in 5% of all infections. So essentially, for the polio virus, you have two vaccines available. Number one is the Salk vaccine. So this is basically an inactivated vaccine, and this inactivation is induced when you treat poliovirus with formalin, and this basically reduces the infectivity. The second option is the oral poliovirus vaccine developed by Sabin. And this uses 3 live attenuated viruses, which doesn't cause neurovirulence or pathogenic activity in the brain to cause all those paralysis effects. And this is done by growing these viruses in monkey cells. And because these viruses are grown in monkey cells, they are suited to attack the monkey host, but they don't necessarily have the features to induce that deadly impact in humans. So that's why that vaccine is quite useful. And we have found that both Salk and Sabin can induce high levels of IgA, M, and G in the serum, but only the oral polio vaccine can induce high levels of IgA in nasal and duodenal secretions. So in some ways, the oral poliovirus vaccine can offer wider protection and a greater antibody response. However, because it is live attenuated, there is a possibility that it can revert to virulence. Although this occurs at a very low frequency, in about 1 in 2.5 million, we still want to be safe, which is why the Salk vaccine or the inactivated vaccine is used more frequently. And essentially, for attenuation to occur in the oral poliovirus vaccine, you can use 3 types of changes, and all of them involve some sort of change in the VP proteins. And it is the type 3 which induces an amino acid change in the 91 residue for the VP3 protein that can easily revert to virulence. Now moving on to the measles vaccine. So measles can actually cause a chronic brain inflammation known as subacute sclerosing parencephalitis. If you Google this is actually a very scary condition, which is why vaccination is important. And essentially in the vaccine, you can get the prevention to these deadly consequences by inducing an immune response to the attenuated form. And we believe that SSLE arises due to a mutant form of measles, which can not be detected by the host properly because it doesn't express structural proteins which can be targeted by antigen-presenting cells within the host. Now, in terms of where we are today, we are developing more viral vectors. We're using recombinant DNA technology to develop virus vectors as well as virus-like particles. And the viral vectors that we like to use include the pox and adenovirus vectors, because these viruses have a very large insert size, which means that we can put in gene sequences for antigens from viruses with greater flexibility. And the goal of these vectors is to be treated so that they only induce a mild infection. And the viral vectors must also be attenuated. And a common one used from the adenovirus is called AdEasy, and it basically allows a shuttle vector containing the pathogen antigen to be inserted. And this has been shown to be effective in the protection against Ebola. You also have some flavivirus vectors being used as well, and they are quite useful too. And the advantages of viral vectors being used as vaccines is that they are immunogenic, which means that they can elicit the high titers of antibody and cytotoxic lymphocyte response. And it also means that there is high viral antigen expression. You can get mucosal route delivery. You can get the viral antigen to be processed and folded correctly. But the disadvantages is that because it's immunogenic and because you're combining different viruses, it is not desirable for the immunocompromised, because this might be dangerous, obviously. But for abortive infections like AVPOX, it is still used. Now, the virus-like particles can be made when you use recombinant DNA technology to clone the viral DNA and then put it into bacterial cells, and then this will generate your virus-like particle. And these behave basically like a virion, infecting cells, inducing the correct antibody and cytotoxic T lymphocyte response. And some of the virus-like particle vaccines include the one against hepatitis B, which basically grows hepatitis B antigen in the yeast, so it doesn't have the genome, so therefore you can't infect, but you would get an immune response against the HBs antigen. And then you also have the HPV or papilloma vaccine known as Gardasil. And Gardasil is useful because it uses this L1 capsid found in the papilloma virus, and this is recombined. And this has been shown to induce an antibody response that is even higher than the one to natural infection. And it also, protects against cervical cancer strains, HPV 16 and HPV 18, which account for over 70% of all cervical cancer. And it protects against HPV 6 and 11, which accounts for 90% of genital warts. This vaccine is introduced intramuscularly at 0, 2, and 6 months of age. And now we have something called Gardasil 9, which has 5 additional genotypes that are associated with cancer. Lastly, we can have a live attenuated vaccine made from genetic reassortment, and this is basically done by using two parent viruses and using their genome to be grown in one cell. So the resulting cell will have the targeted antigen but be attenuated, and the parents might include one that has a virulent antigen and another that is weakly pathogenic. And an example of one which a live attenuated genetic reassortment virus vaccine is called FluMist, which protects against influenza. So it contains genes for the hemagglutinin and neuraminidase enzymes of the current flu strain. Additionally, has about 6 genes which have been modified so that they cannot infect the lungs and only the upper respiratory tract. And this feature is called cold adapted, and this has been shown in inducing the antibody response, the reactive cytotoxic lymphocyte response, and the mucosal immunity, and is even safe in children. But live attenuated or genetic reassortment vaccines are best used for segmented viruses because they allow protection against the assortment of viral strains. So to sum up, whenever you are using an attenuated virus, you would expect fewer doses, longer immunity, that is similar to the natural pathogen response, or maybe even better, such as the case of HPV. But it is not that desirable for the immunocompromised, can be unsafe. There is the possibility that you can revert to virulence as well, and you can also be shed to the environment. Thank you so much for listening today. Have a good day.